Thank you, Claire, and thank you all for the opportunity today to present before the AGRO division of the American Chemical Society. As Claire mentioned, my background is in toxicology and environmental health sciences, and what I want to do today is introduce you to some of the refined safety assessment approaches we take in the what may be broadly termed the pesticide industry, but this would be insecticides, fungicides, herbicides, and then tell you a little bit about some of the advancements that are transforming the toxicology world and how that's impacting our testing. I'll tell you a little bit today. Here's my outline: agricultural chemicals, why we are in this business, what does the regulation of pesticides look like globally, and what's the required toxicity testing. There's a very high bar for mandated requirement tox testing globally that I think will be of interest to people. Talk a little bit about then how we take that toxicology information and put it into a risk assessment process. And this is something we do, again, based on requirements from the US EPA and other global regulatory bodies. And then I'll conclude with some future directions and show you some of the new approaches, techniques that are transforming the world of toxicology and how we assess human safety and risk. So why do we do what we do? Plant protection meeting a global challenge. And on the top three boxes here, you can see we expect world population to grow to 9 billion by the middle of the century. Arable land is constant. This is not typically something that's expandable. And we have a very significant population that are undernourished. And so we, within Dow AgroSciences and within the industry, which is comprised really of about 5 to 6 primary companies, our challenge is then feeding the world. So we do that through two modes here at Dow Agro with agricultural chemistry, again, fungicides, herbicides, and insecticides, each targeting a certain pest, and then through some of our biotech platforms, seeds and traits and healthy oils. And on the bottom, you can see some of these technologies do help to increase crop productivity by 20 to 50%, particularly when you're able to engineer drought resistance, heat resistance into some crops. That can be very helpful. And still, you know, there's a significant loss of potential food to pests annually. So, and you've got some citations on the bottom there if you want to go. These are not Dow Agro statistics or opinions. So a little bit about development of pesticides. Again, I mentioned we and most of our colleagues in the industry are producing agricultural chemicals and also plant biotechnology products. Something may not be known, but we're very similar to the pharmaceutical industry in terms of the resource intensiveness of how development takes place. For us to develop a new active ingredient, similar to an active ingredient in a pharmaceutical, this requires about 10 years and $250 million to bring something to market. The global requirements are, as I said, a very high regulatory bar. Over 100 different studies, and it's really well beyond that, are required to support the health, safety, and environmental assessments prior to us putting something on the market. And this is just a graphic to show you that regulation of pesticides is global. You can see the US EPA on the far left here, but certainly in Europe, where we are under the auspices and have to adhere to requirements of the OECD. European Food Safety Authority is in Parma, Italy. ICAMA is another entity. MAF or JMAF is in Japan, the Australian government, but it's really global. So these are just a few of the major authorities, but there are country authorities as well. In the U.S., let me just tell you briefly about how we regulate things in the U.S. and which laws those come through. Primarily, we're regulated by the U.S. Environmental Protection Agency through some federal laws. These are acronyms. The FIFRA is the Federal Insecticide, Fungicides, and Rodenticide Act. FQPA is the Food Quality Protection Act. This was introduced in 1996 under, I believe it was President Clinton, and this focused specific attention on additional safety requirements for our chemistries for infants and children. The FFDCA is the Federal Food, Drug, and Cosmetic Act. And so beyond the development of, say, an active insecticide, we have to establish what are called MRLs. That's maximum residue limits. So if you were to go to the grocery store and buy a foodstuff, fruits, vegetables, or grains, there are maximal levels of residues that can be in those things. So not only do we have to pass a risk assessment that I'll show you how we do that, there are established maximal MRLs, and that comes through this act. And I won't read you this quote here other than you can just see that the standard we have to adhere to is our products do not pose unreasonable risk to human health or the environment. And I stress the environment because that's become a big component in recent years as we have to make sure we're not targeting ecological receptors as well. So in terms of just some of the, what comes through FIFRA, that's the law that we're regulated under. What are some of the data requirements as we develop a new chemistry? And you can see the studies areas over there. I'm a toxicologist. In terms of the world of toxicology, we have to do at least 28 different studies, and I'll show you the range of what those look like. Re-entry protection, that's example for workers. This is operational people that are working in fields. They can't just treat and then go back in. There's sometimes requirements that there's a 12-hour buffer between when they go back in and treat. Non-target insects. We don't want to be targeting things that are off target. We have to look at environmental fate of our chemistries, what's going on in air, water, soil. Spray drift is another thing. So just the range of study areas, and then you can see some of the study numbers on the right. In terms of data requirements for what we call mammalian toxicology, these are some of the things we have to do on that new active. I will differentiate that from a formulation because just as in a pharmaceutical, if you go buy something off the shelf, therapeutic treatment, it's obviously got an active ingredient in there, but then it's got other things in it as well. Well, we're much the same. We develop an active ingredient, but then we put that into a formulation that's actually used out in the field or on a targeted plant. But these are some of the studies we do. We do a number of acute studies. We have 6 of these, and they cover acute oral, dermal, and inhalation toxicity. And then we're also very interested in primary eye, skin, and irritation and sensitization. We do what we call medium-term studies, 28-day studies using various routes of exposure. We then do what we call a 90-day study. That's often used to help us predict dose levels that are used in what we call our chronic studies. Those are the studies we do to look at cancer-causing properties because we don't want to introduce carcinogens into commerce. And basically, I will tell you that these days the bar is high enough that if we saw something early on in some of our early screening that said we've got a potential carcinogen or a genotoxicant, we're probably not going to advance that chemistry. I won't talk about that today, but it's a whole other facet of developing chemistry and when we make decisions. But why do we do cancer studies in rats and mice? We do those, and we do them for 2 years because the life of a rat is typically 2 years. We want to mimic the life of a human, which is 70 years on average. So, and we do them in 2 different rodent species just to add to the robust database. We do a number of things to look at developmental toxicity. We do these in a number of species as well. We look at reproduction and fertility effects, neurotoxicity, a handful of studies looking at genetic toxicology. So these are just the requirements. They are pretty much standardized globally. There are a few things that some geographies require that others don't, but when we develop a data package, it's intended to be used globally. I've covered some of this, but the routes of exposure, we want to, again, look at varying routes of exposure because we want to mimic what a human might be exposed to. So we do these through the 3 principal routes, which are oral, dermal, and inhalation. There again, you can see the span of time frames we're very interested in acute exposures, but we do these things way up to 2 years in length as well. Multiple species, we don't use primates in our testing, primarily rodents, rat, mice, rabbit. We are developing enough alternative data now that we are hoping that geographies around the world eliminate the need for a 1-year dog study, and that's almost complete, but there's still a few geographies that require that. This does require a lot of animals, but something Dow AgroSciences has been very committed to, and it's one of our sustainability goals, is to actually reduce animal testing by a key percentage, and I don't know, I can't remember if that's 33 or 50% by 2025, but we are very committed adherence to what we call the 3R principles, and that is reduction and replacement and refinement of animal testing. So we have some of the NGO and animal welfare people, PETA, Humane Society of the U.S., these people sit on various boards we have, and they come in annually, look at some of our approaches, and we appreciate their guidance. But we work together in harmony and really targeting our 3R principle. What are some of the areas we look at? I've mentioned some of the basic studies, but we really have to cover all potential endpoints of toxicity in our testing, and that includes neurotoxicology, endocrine, reproduction, development, immunotoxicity. We want to know how our chemistries move through the body and how they're detoxified. So we are very interested in what we call ADME studies, and that stands for absorption, distribution, metabolism, and excretion of that active metabolite or active ingredient. And then finally, we want to look at mutagenicity and carcinogenicity. Within studies, it's like a person going to a doctor. You're interested in getting blood results. You're interested in getting clinical chemistry. So you may give biological samples or specimens. We're much the same in the world of toxicology. These are some of the things we look at within our toxicology studies. We look at clinical observations of the animals themselves on a daily basis. We evaluate all organ systems within the rodent body following termination of the study. We'll do pathology, histopathology on all organ systems. We look at body weight changes. We look at clinical chemistry, and then blood, which is hematology. We measure organ weights. And together, what these, particularly with the histopathology, that's microscope work, looking at lesions. We look at all these things because we want to have a robust picture, and we look for what we call toxicological correlates. In other words, if you're seeing something at a cellular or molecular level, are we seeing that at a organ level? Are we seeing that manifested then, say, in increased organ weights? Because that gives us an idea then, is this effect real, adverse? And from that, we begin to determine how we approach our risk assessment. I won't go over this again other than to show you that in development of an active ingredient in our industry, the tox testing is probably about a 4-year journey. The cancer studies themselves are going to be 2 years just in length of in-life, and that's probably a 3-year study itself. But from beginning to end, the tox testing requires about 4 years of work. Now, from that, because of that high bar, and why do we do all of that testing, we then have to move and use that toxicology information in a risk assessment. And again, this is not something that Dow or our competitors design. We do these based on standard approaches that the US EPA or other geographies would require. So one thing most of you understand, but if this is new to you, it's a very underpinning equation, and that is risk has to involve two functions. One is hazard, and that's what we're trying to understand during all of our toxicology testing, and the other is exposure. A hazard is a measure of an adverse effect, and really it's an intrinsic property, as you can see, of a given substance. But to have risk, you've got to have some element of both of these. I often use I liken an example of a snake, a poisonous snake sitting on your desk in a jar. You've got the hazard there, but if that has a cover on it and you have no exposure to that poisonous snake, there really is no risk. Conversely, you can have a situation, I'll alter that. You could be out in the middle of the ocean where water, it doesn't have high hazard, but if you have tremendous exposure to that low hazard material, i.e. in drowning, or there was a case in California years ago where someone was put up to a test to see how much you could drink. A person actually died from overexposure to a very low hazard material. So that's the same kind of thing we're looking at in risk assessment of agricultural chemistries, is what does that hazard and exposure profile look like? So we take ourselves through a paradigm, and it's called the risk assessment paradigm. Hazard identification is what I just described through the tox testing we do. We then look at dose-response relationships, and I'll show you a dose-response curve. In all of our toxicology testing, we have to do this at least incorporating 3 to 4 doses, because we have to determine what we call a no effect level. We have to test at some low dose that doesn't elicit any effects, and I'll show you why we do that. We also then want to have frank toxicity. We want to see where is the maximal toxicity occurring, and that comes into kind of the exposure assessment and the safety assessment. We also then want to understand, what does exposure to humans look like for you and me in a residential setting, to operators, or in an occupational setting? So we do all of these 3 steps. That comes then down to what we call risk characterization. So we'll do that risk assessment, and then we'll submit this to the EPA. They ultimately are the determinants of whether or not this risk assessment is passable, and whether it then allows us to produce a product label and put this into commerce. So this is an important slide, because how do we take toxicology information and then set permissible exposure limits to humans, or what we call protective thresholds? And we select what we call an NOAEL. That stands for No Observed Adverse Effect Level. And that's one of the that's typically the lowest dose level that we use, but we take it from all those studies we do. We take the most sensitive adverse effects from a toxicology study that really matches a certain exposure scenario. So in terms of the types of protective exposure levels we set acute and chronic ones. And so say we're setting a chronic exposure level. We want to set a level that a human could be exposed to for every day of their life for 70 years. So we're typically going to take that NOAEL from our chronic cancer study, and suppose that NOAEL is 100 milligrams per kilogram per day. Well, we can't just say that's going to be, even though there's no effect at that level, we have to add in some safety or uncertainty factors, conservative or precautionary steps, because we're dealing with humans here. And so what we do, we divide that NOAEL by 10. We call this an interspecies extrapolation factor, because we did our studies in animals, but we're protecting humans. Humans might be more sensitive. So we add in a factor there. We also don't know about interspecies variation. Obviously, every human is different, and so we add in another factor there for sensitive human populations. We then, as I mentioned, in 1996, about 20 years ago, it was mandated that we take a look at protection of children. You know, do we need to add a default 10 in there? And so oftentimes, at the end of the day, we might be adding in an uncertainty factor of 1,000. So we divide this NOAEL of 100 by 1,000. That then becomes what a chronic reference dose is, and that's defined as an estimate of a daily oral exposure to the human population that is likely to be without an appreciable risk of deleterious effects during the lifetime. So that's basically then how we set that safe dose level for humans. And this is just an example of a dose response curve. As I said, We typically are setting doses. Say we have a dose of 0.1, 10, and then go on up from there. We want to see where effects occur. And so on the x-axis here is your dose. On the y-axis is the response. We want to begin to see where do no effects occur. And then, as I just described, we take that level and then divide it by a number of factors to ultimately end up to what I said was a reference dose. That's the permissible or safe dose to humans. So let's move to a little bit on exposure. What is exposure assessment? We really want to understand how people are exposed from all routes. And so we want to understand their potential exposure from air, water, soil contact, and indirect routes. Food chain, there could be very small residues within foodstuffs, and so, we want to take account for that. So it's really taking a robust collective view of what a person's exposed to. That's what exposure assessment is. Here is just a graphic of what is residential, meaning you and I in our homes. What does that look like, and how a person might be exposed to, say, rose dust or something they're using in their home. We want to look, what's the potential exposure from air through inhalation, dust, soil, inadvertent contact through the skin, and then, potentially hand to mouth. We have to be concerned with children playing in yards, on lawns. What does that hand to mouth oral ingestion look like? So I'm just showing this because this is one of the types of risk assessment we have to do. It's called residential. Another one is drinking water. EPA requires us to conduct drinking water assessments as part of what I'll soon describe and show you a slide on, what we term aggregate assessment. And this includes both ground and surface water. So again, we have to assume that our active ingredient is in, both phases or both types of water environment. In this case, a tiered approach is used. We first look at what the environmental fate and use pattern of the product is. We then will conduct, what we start with is a conservative approach called Tier 1, where we will model ground and surface water to obtain estimated drinking water concentrations. We'll feed those into the risk assessment and see if, look, are these levels within a safe exposure scenario, or is some mitigation required before EPA passes the assessment? And those mitigation efforts could look like we've got to decrease how much can go on the land, for example. A Tier 2 assessment would be a higher level assessment. That would be where we would actually do actual monitoring of water sources to obtain, and it's really not estimated, we are actually going to obtain actual drinking water concentrations. So these can be very expensive and extensive studies to conduct because you're actually out monitoring sometimes regionally or nationally to get a sense of what water concentrations look like. What's another type of assessment we do? OPEX really stands for operator exposure. Again, we have to look at folks that work and scientists, stewards in occupational settings that use our chemistry. So we do this obviously for company stewardship reasons, regulatory requirement. It is a default that we have to do. We do these for new registrations. EPA every 15 years or more frequently requires us to, or they will re-examine our chemistry to say, okay, have you done all the tox testing that's now required from, say, 15 years ago? So that's called a re-registration. So we'll take a look at that point in terms of what does the risk assessment look like. And then other reasons we do this, obviously we have to comply with EPA questions, product support, and our response to reported adverse incidents. So just to say that this is a whole other population that we're interested in, and we do risk assessments all the time required by EPA to support product registrations. So what is aggregate risk? This is the combined risk associated with exposure to pesticide residues, really from all potential sources: raw, processed agricultural commodities. Again, we talked about drinking water, and then residential locations as a result of an application of a specific pesticide. So you could have an occupational worker whose livelihood involves hours of work with insecticides. That person, though, also, we have to take into account they have a residence. They may be using chemistries there. Also drinking water. What about potential exposure? So that's what we mean by aggregate risk. Cumulative risk is yet another type of risk that we have to look at. Under the Food Quality Protection Act, EPA said that we must consider cumulative effects resulting from exposure to multiple chemicals with a common mechanism of toxicity. So this is to say that I'll give you one class, and one of them is down here, carbamates or triazines. These act through, like the organophosphorus compounds, they act through the same mode of action. Not each of these classes, but organophosphates work through inhibition of cholinesterase. So what if a person is exposed to 3, say, cholinesterase inhibitors on a daily basis? We have to take that into account and look at the potential cumulative effect. And so that's what we mean by we have to look at cumulative risk assessment. So that's a brief, taking you further through the first couple of parts of this webinar today. I want to conclude by taking you through 6 or 7 slides on what I call tox testing in the 21st century. I've talked a lot about traditional tox testing in rodents, but this was a vision and a strategy that the National Research Council put forward 10 years ago, basically talking about, and you can read it, that there's a lot of advancements in new technologies and approaches that may eventually allow us to make tox testing quicker, less expensive, and really, more importantly, directly relevant to human exposures. And so this has really created within the discipline of toxicology a transformation. I will say we still have to do all the testing that's required right now, but we're in that transformational phase where we're looking at alternative approaches, and the US EPA is now beginning to say, yes, there's enough data, and these are sensitive, predictive enough approaches that we can substitute in some of the simpler studies, such as the acute oral, the skin and eye irritation. We now don't have to use animals anymore. We can use some of these approaches. So what are some of the challenges and opportunities? I would say in terms of some of the opportunities, these are bringing forward in vitro or cellular tests alternatives to animals. We have the ability to do high throughput screening methods now. We can bring concepts such as systems biology, and we're looking at things what I call way upstream. We're not just looking at, say, lesions in animals or descriptive toxicology. We're looking at proteomics, genomics, toxicogenomics to really map tox pathways out now. Computational modeling, this is another, in silico or computer way to do some of this work. And then tox testing of mixtures. This has always been a challenge for the world of toxicology, is how do we test mixtures? Well, there are some approaches now that we're beginning to get a handle on, because people are not just exposed to single entities out in the environment. Conversely, what are some of the challenges we see globally? Well, one is, I'll go to the last point, harmonization. As I said, there are differences in country requirements. So harmonization of these approaches is something we have to work across with various regulatory bodies. Certainly adoption of the novel science into new testing paradigms. And this requires multiple stakeholders. It involves adoption and approval of these, not just by the industry, but by those who regulate us, the regulatory bodies, but also other stakeholders such as the academic community and the NGO community. It's a conversation, and we have to move forward in stepwise with all of these before these things are adopted and embraced. And certainly the point two here, we've got to be very sure that the validation of these new methods, that's in place. We can't just put forward something. We have to ensure that it is predictive and that it's an appropriate conservative surrogate for an animal study before putting it into guidelines. I wanted to give you one example of how Dow has advanced some of our testing in this space, because we did this a few years ago, and we were somewhat pioneers in this, where we said we're very interested in reducing animal testing. So instead of just doing a traditional 90-day rat study, let's begin to do what we call integrated testing, and let's see if we can incorporate 3 other required studies in that one single study. So we embraced and brought in the immunotoxicology study, the neurotox study, and the genetic tox study, all within this one 90-day rat study, and we covered all endpoints. What did that allow us to do? Really, we're hitting our targets for our adherence to the 3 R's principle. We did 1 study instead of 4. We reduced animal reduction from 250 to 90, and we also, that enabled us to gain some early insight on toxicological profiles and targets, which then inform subsequent testing. So it's a far more efficient way to do testing. So, and this is a study that, was accepted or is accepted by the US EPA. So kind of a win-win on all fronts. A few more slides. What's some of the vision of, I'll just say for Dow AgroSciences, from what we used to do in testing to what we do now? We used to do historical high-dose testing, and MTD means maximum tolerated doses. We don't like to force, and we're cognizant of animal welfare, so we like to move away from what we call maximum tolerated doses to more humane doses, and so we have now embraced what we call KMD, which is kinetically maximal dose. So in all of our studies, we employ what we principle called, we collect toxicokinetic information. We want to know how a material moves through the body, and we don't want to do something that's way beyond the linear range. And so that's, I won't go into the concept other than if there's something that's a body couldn't handle, we know what that top dose is, so we've employed a KMD approach. We've gone from just doing descriptive toxicology to we want to understand what the mechanism is or how a chemical acts. We want to move beyond guideline-only methods to what I just described in terms of integrated testing and understanding, again, mode of action. We have to know how something's operating. It's not enough just to know that we're ending up with a certain effect. We want to know, and that leads me to my 4th point here, instead of just assuming human relevance, we understand mode of action, and we'll do extensive, sometimes it takes years of research to determine the mode of action, because oftentimes in a rodent that mode of action is not relevant to humans. But if it is, we want to understand that and then put that into our risk assessment. Sometimes we used to just do default testing. We now have approaches where sometimes we can bridge data, or we can use other approaches to waive a study but still be protective. A couple more slides. What are some of the predictive technologies that we use to inform early phase decisions? And so now I am going to give you a little bit of insight into what we do prior to getting into our required regulatory studies. We're thinking sustainability, not just registerability. We don't want to develop chemistry that has high persistence or is bioaccumulative in ecological receptors. We don't want to develop PBT, in other words, persistent bioaccumulative or toxic chemistry. So we're developing, very interesting early on in developing greener chemistry. We want to understand metabolism and fate. Again, not only in mammals, but we want to know what's that profile look like in ecological receptors? What does it look like in the environment? So we're doing a lot of high throughput screens now. We're doing a lot of in silico, i.e. computer. We're bringing in tools here where we're looking at chem informatics. GastroPlus is a licensed tool looking at, it gives us much better refinement for what that chemistry looks like inside the body. DART is developmental and reproduction testing. We have a decision tree whereby we get an early view of what do the DART properties look like for this chemistry. And then biological profiling. This is something where we're going way upstream. In other words, down to the molecular level, and then we're trying to compare and contrast what do the up and down regulation in terms of genomics look like related to traditional toxicity endpoints. Some of this I've covered, but these are some of the bridging tools and approaches that we will continue and the industry will continue to use as we make this transformation to use of more and more alternative approaches. QSAR is quantitative structure activity relationships. We now can do chemical read across, and this is a big and popular approach used in Europe. Again, in vitro, in silico, high throughput technologies. We've talked about omics, pathway analysis. AOPs is another concept that's been popularized, particularly in Europe, adverse outcome pathways. We want to not just understand the end game or apical endpoints, we want to understand what those pathways look like and what are the molecular initiating events. PBPK-PD, that's physiologically based pharmacokinetic, pharmacodynamic. That's a model where we build that based on animal data, and then we use those to gain insights into predictions for humans. Virtual organs. There are now virtual organ systems that people are beginning to use. So the challenge will be, and the hope is that we'll validate and eventually accept some of these new technologies for tox testing. And finally, just how does this all come together in terms of advancement in safety assessment? It's really refinement to both toxicity testing and exposure modeling. An example here is how might we use biomonitoring data? The CDCs and other government agencies, they collect data on biomonitoring, not just of agricultural chemistry, but things such as cigarette metabolites, all kinds of pharmaceuticals. And so we have a profile of what that looks like for people nationally. How can we use some of those modeling data to back inform safety assessment? We've gone to an approach using what we call chemical-specific adjustment factors. These are actually based on actual data versus what I showed you before, those 10 by 10 by 10 default values. So let's actually use data based on the chemistry versus default values. The TTC approach is another one that's gaining some acceptance, threshold of toxicological concern, really used for things not like active ingredients and pesticides, but for things where there is, where we're working with agents that don't have a high hazard profile. And then bioactivity, move your mind from just toxicological endpoints to bioactivity. We are actually looking at that as a point of departure versus actual apical toxicological effects. And this finally has all been brought together. There are a number of multi-stakeholder collaborators, again, involving all stakeholders that are pushing forward concepts of Tox 21 and Risk 21. So with that, I think I'll pause. I know that's a lot to cover, but I want to thank you. And at this point, I'd be very happy to take any questions.